senotherapy. What does senotherapy actually aim to do for not only the skin, but more systemically as well in terms of anti-aging? And how do antioxidants play a role? And they play a very important role. So join me for this presentation. In order to start, senotherapy really begins with our understanding of aging. What is aging? And I present this slide because it really tells us the difference between what we normally consider aging. We call that chronological aging. So that is simply the number of years that has passed since you were born. And then there is biological aging. And biological aging is really related to how old are your cells. And those two things are not necessarily the same. You could have chronological age where the age of your cells far exceed the age that you are chronologically. So we call that advanced aging or premature aging. But then also the inverse is true. You could have slower aging cells, which means that the cells are more healthy, they're more active, and they are truer to their form. And this could be irrespective of the actual age of the individual, and we call, and that is more delayed aging. So this tells us that there is room for us to work. In other words, there is a therapeutic window where we can intervene and increase the activity of cells. And that's going to mean that the cells behave in a more youthful way, irrespective of the age of the individual. And if cells can behave more youthfully, then the overall appearance to the skin structure, because these cells, particularly dermal fibroblasts, they have a really important role in maintaining the structure of the skin. And if those cells function optimally, then the appearance of aging, and by all means, the actual age of the tissue itself, is no longer in line with chronological aging. So let's bring us back to those dermal fibroblasts. And I appreciate that most people haven't seen in real life what a dermal fibroblast looks like. And so on the right-hand side of the screen is a short video, and it's a video showing the dermal fibroblast in its appearance. It's long and it's striated, which means that it's kind of skinny and slender. And it's moving along these collagen fibers. You could see if I press play, how quickly these cells move. And they're really hypermobile cells. They move around collagen. And when they move around, they're constantly making and degrading collagen. That's their main role. They're not just collagen, but elastin fibers, glycosaminoglycans, or otherwise known as the Gu molecule. Fibroblasts make up all the extracellular material that makes up the structure and tissue of the skin. And so it's important to remember that they're not only just making these things, they're also degrading the old collagen. And so continuously they are renewing the skin surface. And when they have a good balance between making and degrading, this balance is called homeostasis. Aging can flip this balance across, and I'll show you how that looks like. But before that, I'll show you what the skin structure looks like. A young, healthy skin has really healthy collagen. The collagen here is laid parallel, so we call that, in an organized way. Just beneath the collagen, there's also elastin, and you could see that the elastin is interlinked between other elastin molecules. That gives it its spring-like effect because elastin's got this kind of elastic band effect, whereas collagen is more tensile, so it resists the movement of being pulled apart. So it gives the skin its firmness structure. In older skin, where elastin is being produced in a way unlike young skin. So you could see that the elastin fibers are thicker. There is less cross-linking between those. So that spring-like effect is lost. And even the collagen fibers, they're no longer arranged in a very neat way. And this has drastic effects to the skin. So it goes to show that it's important. There's been a lot of papers recently, 2018, where a major article has landed itself in the Science Daily. And the article showed that dermal fibroblasts, when they age, they lose their identity and they no longer behave like dermal fibroblasts. They actually behave more like fat cells. And so they've called them a cell type with a lost identity. And since then, more and more research articles have demonstrated that dermal fibroblasts lose their function with time as they age, and that certain interventions can revert them back. So we know that, sure, aging, biological aging, can cause fibroblasts to lose their function. And if they lose their function, it causes the effects of aging on our skin. But there is room for us to intervene. And this is a whole bunch of new research has just is changing the game in this area. So coming back to what aging dermal fibroblasts look like. Well, these are dermal fibroblasts here, right where my arrow is pointing. You could see that they're shorter cells, they're less numerous, they actually have more fats accumulated. And if you look at this micrograft here, on the left-hand side, you could see collagen in young skin. And note that the collagen is orientated in a parallel way. It's very organized. It's laid down in a very deliberate way. But in older skin, the collagen is rather clumsy. It's not laid down in any particular way. It's kind of all over the place. And the collagen fibers are a lot thicker. And it's because the fibroblasts, which age, don't do a very good job at producing collagen or degrading old collagen, and it leads to the effects of the skin. Now, let's put some words to the concepts that we just described. So this is where senescence comes into play. Senescence describes the very nature of this process. And we describe senescence when the cells have lost the ability to divide and to function. And that's exactly what happens to fibroblasts when they reach a critical stress point. You could see that this fibroblast, which is now in a process called senescence, looks like the fibroblast that I showed you earlier in a previous slide. We'll go back to this. I'll try to go back to that slide. No, I can't do that. For some reason. Oops. Yeah, here we go. That looks like this. So we could say that these fibroblasts are senescent fibroblasts and these are non-senescent fibroblasts. And it's also important to remember that senescence is often an irreversible condition. So once fibroblasts get to a point of senescence, they are no longer able to revert back into their youthful state and be highly functional. The question is, what causes cells to become senescent? And there are two main factors that causes it, but it is mostly damage and stress. It has very little to do with age often. And here are the reasons why. The first point is called telomere attrition. And it is where biological aging can push cells into senescence. And let's just describe that. When cells divide, and this is a picture of DNA coiled into a structure called a chromosome. This is rather a chromatid. And when cells divide over periods of decades, every cell division, a little bit of DNA is lost. And that's because cell division isn't a very complete process. And this is a normal occurrence. But when the DNA is lost to a certain point, we call that the critical threshold. It signals for the cell to become senescent. And this is one reason why cells become senescent, simple aging. Now, it's not the complete reason. I'll come back to this point later. The majority of senescence doesn't come from biological aging. It comes from stress-induced premature senescence. In other words, cells are exposed to stresses like UV radiation, UV exposure or irradiation, oxidative damage, DNA damage, periods of hypoxia, or otherwise known as loss of um, oxygenation in tissue, or dysfunction of certain components within the cell, like mitochondria. These are all known to cause senescence. Here, the example, these are the triggers. The normal cell is experiencing an abnormal amount of stress, and therefore the cell becomes senescent. And when a cell becomes senescent, not only does it no longer divide and no longer become active, but it also causes other effects. So this is the hallmarks of what senescence looks like. So sure, cells no longer divide. There is damage to components of the cells. But again, those senescent cells don't just mind their own business. They produce a secondary phenotype, which is damaging. And this is called the senescence. Oops, that went too far. This here is called the senescence-associated secretory phenotype. And these molecules that are secreted only by senescent cells tell the other cells around them to also become senescent. And that's a really critical point because it induces a secondary senescence. So you kind of get this propagation occurring where senescent cells cause other cells to become senescent. So, the reason why I bring this point up, it's really critical because what we do in the clinic, and in the clinic, I can pretty much summate the things that we do in the clinic into two categories. One are these techniques that we apply. And these are a few. I appreciate there are many more techniques, but these are just some of the techniques. They're microneedling, SQT, IPL, chemical peels, and there's a longer list. Or sometimes we use biostimulators like peptides and retinols, vitamins and antioxidants. And sometimes we do a combination of those two, the microneedling or the SQT or treatments with those biostimulators. All of those things that we do to stimulate fibroblasts so they can produce collagen and they can produce elastin. That is the cornerstone of many of the techniques that we apply in the clinic. Now, the reason why I mentioned that is that our techniques are only as effective as the health, the pre-existing health of the fibroblasts that you're treating on the skin of the surface of that individual. So I use the analogy, it's like using high octane fuel. And if you apply high octane fuel to a Toyota Corolla versus a Ferrari, you're going to get a very different response. In other words, someone who has healthy fibroblasts may respond very well to these certain techniques, all these biostimulators. Whereas someone who has fibroblasts which are senescent due to stress, well, the response that you get may be low or moderate. So I think when we think about treatments, we usually start from this top-down approach, techniques and biostimulators, but we really ought to be thinking about how can we improve the function or the health of these cells so that they can then be responsive to the treatments that we apply. And that is really the way forward. And so how do we do that? Well, we always start with how we can prevent senescence first, and then we'll look at what we can do clinically. So when it comes to preventing senescence, because senescence can be caused by two ways. Remember, we talked about the biological senescence, the telomere attrition. There's still a lot of questions we don't know. So what age does telomere attrition occur? And that's a big unknown. And the reason why it's an unknown is because it's highly varied. Your diet and your lifestyle will dictate when that occurs. And those who have poor lifestyles or poor diets with a lot of stresses, they may reach telomere attrition faster. There is a lot of good evidence that individuals who take vitamin D supplements, they can actually elongate those telomeres and they could offset senescence. and those who have diets rich in certain antioxidants. And this particular antioxidant called carotenoids. These are found in red or more colored vegetables like carrots in particular, red capsicums, and the sorts. These are known to prevent the shortening of those telomeres. So this is why biological aging, although it is one of the reasons why, it's a highly varied reason. So your lifestyle will dictate that more. And certainly there are things we can do to prevent it. What can we do to prevent the largest cause of senescence, which is the stress-induced premature senescence? Well, we can intervene here very easily by repairing DNA damage. And I'm not going to focus too much on this point. I did speak about this at a different conference, and I will mention some points, however. In short, coming back to this slide where I showed that stress-induced senescence is mostly caused by these triggers damaging and stressing abnormally normal cells. And the link here is causing DNA damage. When cells are stressed, the DNA becomes damaged. And if the DNA is damaged enough, it can trigger cell senescence. So what can we do now if cells do become senescent? Because often you reach this point anyway. And this is where senotherapy comes in. Senotherapy is the only therapy where we actually targeting senescent cells. And we're doing that for a few reasons. And this is a nice diagram that shows you where senotherapy comes in. So let's go back through this diagram. You have normal cells. These cells experience stress. And the stress can cause the cells to become senescent. Remember, senescence is a one-way process. Once cells become senescent, they can no longer revert back to being a normal cell. And those senescent cells accumulate and they can cause harmful effects. Of course, they're going to degrade the tissue, but cancer is amongst other things that they can cause. Senotherapy aims to either do two things. One, kill the senescent cells. And that's really important because if you want your tissue to recover, you need to remove the senescent cells for normal cells to regenerate. I'll discuss that more. Or the other is to block the signals that those senescent cells secrete to neighboring cells so that you don't get an enlargement, an accumulation of senescent cells. Now, fortunately, antioxidants, and there's certain types of antioxidants, in this case, are polyphenols. They have a well-defined role in senotherapy. In fact, they are one of the major categories that have been studied as possible senotherapeutic targets. So let's hang on to that and discuss that more. The polyphenol mechanism of action during senotherapy. And we have to think about how do senescent cells behave? Well, senescent cells behave in a very similar way to cancer cells because both senescent cells and cancer cells do one thing in common, and that they avoid dying. They avoid cell death. And the only way that they can do that is if the cell upregulates these pro-survival pathways. And these are three major pathways that I've summarized: the PAKT pathway, the MAP kinase pathway, and the BCL2 pathway. And what's interesting is that plant-based polyphenols, these antioxidants, and I've listed a few here, they have been shown to inhibit these pro-survival pathways. And it allows the senescent cells to die. And in a similar way, it can allow cancer cells to die. So a summary of the types of senotherapies are these two. One is the senolytic therapy, which has received the most research. And this is where the approach is to kill senescent cells. And if you do that, then you make way for healthier cells to grow and regenerate. And when healthier cells regenerate, the tissue can eventually regenerate. And the other is a xenomorphic form. And this xenomorphic, although less studied, is still equally important. It silences the senescent signals, the SASP molecules from senescent cells, and therefore it prevents the secondary senescence. Let's look at the senolytic therapy. So here's an example of tissue. And in this tissue, it's experienced stress, and some of the cells in this tissue have become senescent. Now, if you recall, I mentioned that senescent cells don't just mind their own business. They produce those secondary secretory factors, right? And those instruct the other cells, which weren't originally affected, to become senescent as well. We call this secondary senescence. Secondary because they weren't there to begin with. They're only senescent now because the first senescent cells instructed them to become senescent. So senolytic therapy aims to kill off all senescent cells, to clear them from the tissue. And at the end, the tissue looks a little bit like this. But that's fine because these cells have regenerative capacity. Healthy cells can divide. And eventually the cells regenerate and the tissue is saved. And that is the approach of senolytic therapy. Whereas xenomorphic therapy does something a little different. It doesn't directly kill the cells, but once the cells are stressed and become senescent, it blocks the production of those senescent molecules. So it stops those cells from making other cells senescent, and that's equally important. But when we think of how we apply this, well, senolytic therapy is designed to be applied in an intervaled application. In other words, about 2 weeks. That is the amount that is described in the clinical trials that have been done in senolytic therapy, anywhere between 5 days to 2 weeks. And so you apply the treatment for this period of time, a short period of time. Hopefully the senescent cells die, and then you wait for many months for those new cells to regenerate and to restore the tissue. Xenomorphic therapy requires continual application daily because you're blocking the signals. So the signals will always be produced. So you have to just always block them. So it requires a continuous application. Now, the question here is, what are the compounds that can act as senolytics, and what are the compounds that can act as xenomorphics? And I've taken this image here, and these are the ones that have been investigated so far. And I can categorize them into 3 major classifications. These are either antioxidants in this list, and I've gone through the liberty of highlighting the ones in a green star. These are naturally occurring antioxidants. And the others are cancer drugs, and the others are antibiotics. So you should ask yourself, why are cancer drugs used as senolytics? And if you recall, I mentioned that senescent cells behave much like cancer cells. They prevent dying off by upregulating pro-survival pathways. So cancer cells or anti-cancer medication, particularly the first one here, is been shown, in fact. And this is what I would like to focus on in this particular part of the presentation. It is the first reported senolytic combination combining a naturally occurring antioxidant called quercetin with dasatinib. And this unique combination is the first to be described only very recently, in the last 3 years, as being more effective than any single compound alone. It causes pro-apoptotic it causes the apoptosis or the cell death of senescent cells. So this FDA-approved anti-cancer drug, it kills these senescent cells by inhibiting a tyrosine kinase inhibitor. And quercetin, again, a naturally occurring flavonoid, which is another kind of polyphenol, interacts with the PAKT pathway and the BCL2 pathway, and it causes apoptosis. And again, it's important to remember that the research here has shown that this two combination works better, is more effective at targeting senescent cells than any of these drug combinations alone. And that's the take-home message. Now, the problem is, before I get to the problem, I'll just summarize these. This combination has been used so far in 5 clinical trials, and these clinical trials all have one thing in common, that the disease pathway is caused by has a major role with senescent cells. So they're trying to kill senescent cells in order to regenerate tissue. And some of these have had really great phase 1 and phase 2 and have moved on to phase 3 clinical trials. And these are ongoing. In regards to xenomorphics, well, there have been no clinical studies on xenomorphics to date. But there have been animal-based studies on xenomorphics, and I've concluded the results of a few, mostly phenicin, which is performed on these keratocytes, but there are other types of antioxidants, and they showed pretty good effects as well. The reason why there are probably no clinical studies is because of its continuous nature. You would need many years of continuous application of xenomorphic therapy to produce, and then simply to fund those kind of studies is very difficult. So it's not that they don't work, or it's just that it's just a difficult study to fund. Okay, so coming back to the combination of dasatinib and quercetin, it's a pretty difficult ask to tell a client to take an anti-cancer drug to improve their skin health. And so a paper that was released months ago by a German institute, a medical research institute, and really asked the same question: is there a naturally occurring alternative to this anti-cancer drug that can be used in combination with quercetin as a senotherapy? And it was a very exhaustive study, and they have identified only one compound that was similar. It had a similar gene expression induced by the anti-cancer drug, and that compound is derived from pepper long roots. And this is the compound here, pepper longumin, and it is the only compound that's been identified to work in a similar way. The study also identified 3 other natural xenomorphic compounds, but these were xenomorphic compounds and were unlikely to work in combination with quercetin. So really, the only viable option that we could use in the clinic is the combination of quercetin and pepperlongumin as a novel senolytic combination. And this will be the first of its type. Unfortunately, there is no product on earth that contains these two at doses that are therapeutic designed for this. This is something that we at Redox Skin Lab have proposed moving forward, and that the scientific community has overwhelmingly supported. I'll talk about that a bit more. Okay. Antioxidants as polyphenols in xenotherapies. Well, most natural polyphenol xenotherapies, such as phenicin, quercetin, and resveratrol, they're all very useful, but they have their limitations. One is that they have limited stability and they have some toxicity when taken orally, and they tend to be have a low bioavailability when taken orally as well. As the problem. But I think the problem with these compounds, particularly quercetin and phenicin, isn't that it has very little to do with the problems I mentioned earlier, but it has mostly to do with the coloring effects. Phenicin and quercetin induce strong coloring effects to the skin, and I'll demonstrate that. And then in this particular slide, we have shown in others as well that quercetin, when combined with zinc oxide, produces a unique jaundice-like effect to the skin. You could see the yellowing here. It's very unpleasant. And thus, it just cannot be used with physical sunscreens that contain zinc oxide. and the reaction occurs in mere minutes. So, how does the reaction occur? And if you're enjoying science, and I like me, you might be curious. Well, here's a quercetin molecule, and I've used a little a red shape here just to identify that. What is really well described is that zinc is able to link two quercetin molecules together. This is called the quercetin zinc complex. Um, but when a zinc oxide, however, contains this oxygen group, looks a bit different. Zinc oxide has a lattice structure, intentionally designed, and it's why it's actually used for as a sunscreen. So when you apply when you apply zinc oxide to quercetin, well, you have this quercetin zinc oxide lattice forming, and so you're able to propagate the orientation of the quercetin within the structure of zinc, and it causes the color change. And if you don't believe me, here is some more research that shows this effect. Here is on the left-hand side a vial of quercetin on its own. Appreciate the color here, and you have zinc, which is clear. When you mix them, immediately there's a color change, which is a light yellow. And over the course of a few minutes, the color becomes more and gradually darker. And over a mere 10-minute period, you have a deepening of this color. And what's occurring at the molecular level is that quercetin is being complexed with zinc sulfate. And so you're getting this growth occurring of this product. So this is what I showed earlier, zinc oxide lattice with quercetin embedded. And then as the reaction continues, well, simply more of this occurs. The lattice expands and it expands and expands. And as it grows, the intensity of the color increases. And so this is what we mean that we cannot use sunscreens with quercetin. Here's an example that we've done, showing you the application of quercetin only after a few minutes with zinc sunscreen. So this was a quercetin-containing solution at the right therapeutic dose, and sunscreen was applied that contains zinc oxide, and this was the reaction. Well, is it a problem that most people that this reaction occurs? Why hasn't anybody else noticed it? Well, this is the question that I had asked myself. I did a quick search on the EWG Skin Deep database, which contains over 110,000 products, skincare products. Less than 5 serums contained quercetin, and the doses that were present were undefined. And so we suspect that the reason why this has not come to the attention of the skin world is because there are very few products, if any products, that contain quercetin in serums. And if they do, they probably don't contain enough or the functional dose of quercetin to cause a meaningful effect on the skin. And we have deduced that the functional dose of quercetin is around 7 micromoles or between 5 and 10 micromoles. And that was based on in vitro studies, topical studies. Now, to use these doses, you most definitely will get a reaction if you use the incorrect creams that follow that. And we will propose a solution to this. So coming back to the clinical trials for dermal senotherapeutics, well, I did show you that there were 5 clinical trials for senotherapy or senolytic therapeutics, but none for dermal. And the reason why is that one, we probably just haven't gotten to it yet. I suspect there will be clinical trials in this area. But scientists are still debating a few things, whether it's topical versus oral, whether it's limited, the stability, the oral route, and a few other points as well. And I make the assertion that topical application is always better because you can achieve the correct dose topically than you would orally. I can go into that later, however. Okay, so to summarize this particular talk, the take-home message here is that cellular injury is the largest contributor to premature senescence and in dermal fibroblasts. And when dermal fibroblasts become senescent, they cannot turn back. And in fact, those cells that become senescent, they resemble fat cells more than fibroblasts. They lose their functionality, and it is the first step towards aging skin. And then you have the problem of secondary senescence, which may ensue as well and further propagate and amplify the effect on the skin. And so often, again, I'll point out that the things that we do in the clinic, the techniques and the biostimulators, they're only as effective as the health of dermal fibroblasts. So we should be thinking about how can we improve the health of fibroblasts so that they are more responsive to the treatments that we deliver. And this is where senotherapy comes into play. It should be a word that is talked about daily in clinics, senotherapy, removing senescent cells, undergoing treatment to remove senescent cells. Now, fortunately, polyphenols, antioxidants can remove senescent cells, and we propose, along with the scientific literature, that quercetin and pipalongumin are a novel potential senolytic combination. And at Redox, and this is just a side note, we are developing the world's first dermal senolytic therapeutic protocol with the combination of quercetin and pipalongumin at functional doses in a topical serum. And this will be applied in an interval process, so a 2-week application, an evening application to prevent the concurrence of zinc oxide, to remove these cells. And I'll be interested in chatting with clinic partners who will be, really the new frontiers to this type of treatment. And just the last slide about Redox, where we started, what we stand for, and our research focus and safety protocol. We started in 2021. And we are constantly in research and development, trying to think of innovative solutions for clinics, users, and ultimately to benefit the client and really be the pioneers in this area. So I hope you enjoyed that presentation, and I'll thank the organizers for the opportunity to deliver this. And I'm also happy to even answer any questions if you reach me directly through my email address. And so thank you very much.